Good morning, everyone, and thank you for joining us for today's webinar. My name is Josh Vickers with RTI International's Forensic Technology Center of Excellence. I will be your host and moderator for today's first webinar of our National Forensic Science Week. Today's webinar is entitled Mixtures, Microhaps, and Modeling, Oh My! Cutting-Edge Research in Forensic DNA Analysis from Early Career Scientists. Today's presentation are early career researchers that were awarded FTCOE sponsorships to the prestigious Gordon Research Conference and Seminar. They will present novel projects in forensic DNA analysis. And then once we are finished with everyone's presentations, we will have our Q&A session at the very end. Just a quick disclaimer, the opinions, findings, and conclusions or recommendations expressed in this presentation are those of the presenters and do not necessarily reflect those of the Department of Justice. Any products and manufacturers discussed are presented for informational purposes only and do not constitute product approval or endorsement by the United States Department of Justice. We have 6 presenters today. We'll move into our first presenter. Our first presenter of today's webinar is Ms. Tiffany Lane, and she will be presenting on the development of a novel isothermal amplification panel for cost-effective and rapid body fluid identification. Ms. Lane is a chemistry PhD research candidate in Dr. James Lander's laboratory at the University of Virginia, working on NIJ-funded research in body fluid identification methods. Before joining the Microfluidic Analytical Chemistry Lab, she completed a Master's in Science and Forensic Science concentrating in forensic biology, and then her Bachelor's in Biology and a Bachelor's in Forensic Science from Virginia Commonwealth University. There she completed research determining environmental effects on body fluids and body fluid identification with Sarah C. Schultz-Williams and Dr. James Robertson. She has presented at multiple conferences, including the American Academy of Forensic Science, PitCon, and the Gordon Research Conference, where she was voted co-chair for the Gordon Research Seminar in 2020. Now I'd like to go ahead and hand things over to our first presenter of the day, Ms. Tiffany Lane.
Thank you, Josh, and thank you to RTI for this opportunity to present my research on body fluid identification. And so jumping right in, there are many different methods that you can use to identify what body fluid is on a piece of evidence, but they all do the same thing, which is exactly that. They tell you which of the various fluids that you see on the screen that we secrete could be on a piece of evidence. So you take a swab or a cutting, put it through this method, and you get one of these out. Some of these body fluids also need to be deduced further because of their similar nature, like venous blood and menstrual blood. And these methods have different advantages and disadvantages. So one, they do what they're supposed to do. They narrow the possibility. They have moderate accuracy and sensitivity. And they've been around for a few decades, so they're very reliable, and the forensic community trusts them, and we know them. However, the targets of these methods are not human-specific, and so that can pose a problem with false positives. Many of these methods have multiple pipetting steps or long wait periods, and they give a colorimetric result that relies on the analyst to correctly identify the color, which brings in a subjectivity analysis or subjective analysis. And then lastly, there's not a fluid for each of the different fluids. There's not a method for each of the different fluids, excuse me. And so these are some of the things that we can rectify. So this is where my research comes in. I am developing a method looking at these 5 body fluids. I am targeting a messenger RNA gene. I use a universal lysis that has been developed and optimized in the Lander's lab. I purify for total RNA using the QIAGEN RNeasy mini kit, and then I perform LAMP, which I'll tell you about in just a moment. This takes approximately 30 to 45 minutes, and then I use image analysis to analyze the colorimetric result. Some of the goals of this research are to have human and body fluid specific targets, allow the least amount of hands-on time possible, have objective analysis, high sensitivity, 1 temperature to amplify all the body fluids, minimal assay time, and of course a full body fluid panel. So what exactly is LAMP? It stands for loop mediated amplification. This is an isothermal method that uses 4 to 6 primers. So at the front, on one side of your gene, you have 2 different primers. On the other side of your gene, you have 2 other primers. And then you can have 2 primers on the curved part of this dumbbell structure right here. How this works is one of your genes will anneal and elongate across your gene. A second primer will anneal and elongate across, performing strand displacement amplification. The same thing happens on the opposite side of the gene, and you end up with this dumbbell-like structure. This is the critical structure for LAMP. From here, you have multiple priming sites that allow for amplification of your target gene and gives you out multiple size fragments with multiple copies of that gene. If you were to look at this on a gel, oh my gosh. Okay. So if you were to look at this on a gel, you end up getting a ladder-like profile as shown, and this is unique for each primer set that you use. This method can take anywhere from 15 minutes to 60 minutes depending on what you're looking at. And so some of the advantages are that it's isothermal, so we can develop simple instrumentation, a heat block. We can use human-specific and body fluid-specific targets. This method has minimal hands-on time, and of course it gives you this unique profile as I showed. For my research specifically, we're using this method with a colorimetric dye. We use the New England Biolabs colorimetric lamp kit, which includes phenol red. This is a pH-sensitive dye. At the start of the reaction, the pH is approximately 8, and so you have this reddish-pinkish color. As a nucleotide is added to the growing chains, the pH, a hydrogen will be popped off, and the pH turns more acidic to approximately 6, and it will give you this yellow color. To eliminate subjectivity, we use image analysis to analyze this. And so we'll take a picture of the reaction, and then using ImageJ software, we will look at the hue of the results, and then we can graph it as you see in these red bars. The red bars show at the start of the reaction what a positive and negative value would look like, and then after 30 minutes, the yellow bars show you what a positive reaction would look like as well as a negative reaction, and you can see that hue has now fallen into the green or positive range. So this method and this analysis method is advantageous, 1, because of its objective nature, 2, we can automate this, and 3, we can analyze multiple samples in tandem. So first thing we wanted to do was look at temperature optimization for this assay. So we decided to look at a variety of temperatures. Here you see on the x-axis 63 degrees, 65 degrees, and 67 degrees. And we decided to continue on with 63 and 65 degrees because of the amplification in the positive samples after in all of the different body fluids. This occurred at 40 minutes. However, we have a target of 30 minutes. So the next thing that we decided to look at was primer concentration. We looked at a variety of different primer concentrations for each of the fluids, as you can see on the x-axis. And the idea behind this was if you have a higher primer concentration, then you have more primers in your assay that have a higher chance of annealing to your target to amplify to get you to that color that you need. We ultimately went with 1.5x primer concentration for semen, saliva, and vaginal fluid to get us to that 30-minute mark. And then for blood, we went to 0.75x due to the sensitivity of the primer set that we're using. From here, we looked at the specificity because any body fluid identification method definitely needs to be specific for the target fluid. And so as you can see, each of the primer sets we have chosen only amplifies the target fluid that we expected it to. However, blood does amplify venous blood and menstrual blood. We are We are in testing for our menstrual blood target, however, which will be the discriminating factor between the two, so we can use those in tandem. Lastly, I wanted to tell you a little bit about the instrumentation we are building for this research. This is our version 1, where it's a acrylic laser cut imaging box. It holds a 96-volt plate on the top, and then you can slide a camera phone into the opening, take a picture, and analyze on a laptop. Version 2 we built was 3D printed. It can hold a 96-volt plate, strip tubes, or a PCR tube, and it has static lighting, and it's also fully enclosed, so it eliminates any stray light. Again, you take a picture with a camera phone, and then we analyze the data on a laptop. Lastly, we have developed version 3, which has all the same features of version 2, and this one's including automated hue analysis, so we have automated data analysis of all of our samples. It includes a heating chamber and fan, so now we don't have to rely on a thermocycler or a heat block to amplify our samples. And by taking a picture and analyzing every minute, we have semi-real-time detection. Just getting into some of the optimizations we had to do for this version 3 instrument. One of the things we had to look at and make sure of is that each of the sample wells are giving us the same temperature and are stable. And so this graph here shows what it would look like if we preheated our instrument versus if we started our instrument at room temperature. And so the orange here shows what it would look like, how long it would take for the samples to get to optimal temperature if we started the assay with the instrument at room temperature. So it takes approximately 1,500 seconds. And then the blue bar here shows that if we preheated this instrument for 5 to 10 minutes, then those samples will reach up to temperature and stay there, stay stable much quicker. So we ended up going with preheating the box. Next, we look at image analysis, because this version of the instrument does not use a camera phone. It now uses a Raspberry Pi camera. And so we run some reactions, and as you see in this graph here, the negative stayed in the negative range or negative hue for up to 60 minutes, and the positive samples turn positive after 30 minutes and stay there after 60 minutes. So right now this instrument is looking very good and helpful for the research that we are developing. So in conclusion, we have developed a LAMP assay that allows for body fluid ID in 30 minutes. We've optimized temperature and primer concentrations, and we've also looked at the specificity of different primer sets for the different fluids. We have built and are in testing of our version 3 instrument that encompasses the entire assay, allows for semi-real-time monitoring and automated image analysis. And then in the future, we are finishing the assay development for vaginal fluid as well as menstrual blood. And then we're going to continue and finish the testing on this instrument, which can be used in a forensic laboratory. This instrument has simple instrumentation. It has a 96-well plate format, but can also hold strip tubes or just a single PCR tube, depending on what you're doing. And this does the analysis of the entire assay for you. Further into the future, we are going to develop a microfluidic device that can be used at a crime scene. It will be able to identify multiple samples and multiple body fluids in tandem. So I'd like to thank Dr. James Landers, my PI, for allowing me to do research at UVA in his lab. I'd like to thank NIJ for funding this research, New England Biolab, specifically Dr. Nathan Tanner for all of his help in this research, and the entire Landers lab, specifically noting Angie Sway, she is my right hand, Aaron, Annie, Antea, and Paolo. Thank you, and hope everyone's doing well.
All right, thank you so much, Tiffany. We are going to jump right into our next presenter. We do have some questions coming in, so we will address those at the end. Again, today's presentation is coming from early career forensic scientists, so we are very excited to be able to give them a platform to be able to present their research on. So our second presenter today will be Miss Anchi Sui, and her presentation will be on rapid screening of human male DNA via colorimetric loop meditated isothermal amplification. Ms. Sui is a chemistry PhD candidate at the University of Virginia. Her advisor is Dr. James Landers as well, a leader in microfluidics. Her research focuses on developing simple, elegant, and portable solutions for unmet needs in modern forensic science. She comes from a molecular biology background and began her career at a biotech startup company in New Zealand, and then moved to the U.S. to pursue R&D in rapid DNA in 2012. Currently, Anchi is part of a multidisciplinary team developing a rotationally driven microfluidic rapid DNA device funded by the Department of Defense. Her webinar will be on a project of special interest to her, which is the development of a rapid and accurate male DNA screening workflow that aims to alleviate sexual assault evidence backlogs, which is also being funded by the National Institute of Justice. And without further ado, I'd like to hand things over to our second presenter of today, Miss Anchi Sui.
Thank you, Josh, for the introduction. I'm very honored to be here to be presenting my research, and I want to thank the RTI and SPCOE for providing this platform. This is great for reaching out to a wider audience. As we're constantly looking for industry partners, we get feedback is always welcome and very valuable to us at the university. So any feedback, any questions are welcome. All right, so for the next 10 to 15 minutes, I just want to expand on what Tiffany already talked about and show you how LAMP can be a useful screening tool for looking at the presence of male DNA, particularly in sexual assault cases. So for this audience, it is probably not news to you that we have a substantial backlog in the U.S. The number is hard to track. The last citation I saw was probably around 200,000 untested kits in the U.S. And these are just some breakdown for each of these states, for these bigger states. And in Virginia, where I live, there's currently over 2,000 untested samples. There are multiple factors contributing to this large amount of backlog, and the one I'm going to address today is the time it takes to analyze them. So let's look at the typical workflow for analysis of sexual assault kits. It typically begins with the screening process, which commonly uses PSA, which is a lateral flow assay to look for prostate-specific antigen. Alternatively, labs can do a microscopy examination to look for the presence of sperm, but that is quite labor-intensive, so PSA is more commonly done. And if that's positive, that sample is extracted for DNA, which can take up to 5 hours depending on your lab, and that DNA is quanted before the downstream DNA profiling can occur. And if that sample is absent, then the analyst has to go back and choose another sample and begin this process again. And you can see the screening process is quite significant, and if that beginning screening gives you a false positive, then the later process is not needed. And so that's why PSA has been criticized for its limited use, because it's a biochemical assay. It doesn't tell you that there's sperm present, and so my proposed method is one that looks for DNA. So to do that, we begin with an enzymatic lysis method, followed by LAMP, which Tiffany has already described, and you go on to produce the DNA profile that way. So, and so that will cut down the screening time to about 3 hours, which is quite a significant saving in time and resources. So this lysis method is quite different from what most people are familiar with the solid phase extraction method, in that this method has no washing steps, no spin columns, and that means there's no loss of DNA. This was originally developed by Dr. Cotton at Boston University, where she used a protease called acrosol, followed by another protease called EA1, which are now both provided by Zygem. So this was originally a two-step method that takes 45 minutes. And in the lab here, we've cut it down to, we've optimized it, so we use a mixture of protease instead, all in a single tube. And in 23 minutes, you can see that the lysis efficiency is quite good. So this is showing 3 different cell concentrations from 10 to 1,000 in these different color bars, and the dotted lines are showing the estimated 100% lysis. And so what you also see here is that without the addition of the proteases, the DNA yield is negligible. What we've also optimized here is that we can decrease the incubation time to less than 10 minutes and still retain the efficient lysis of sperm cells. In addition, the buffer strength has been halved, but still, again, retain that efficient lysis. So this was done to keep compatibility with the downstream processes. So since Tiffany has already walked through LAMP with you, I will just go over it very briefly. Obviously, it's an isothermal method, uses up to 3 sets of primers, and the major difference between her and I is that I use a metal indicator, so in this case, it's called hydroxynaphthol blue, which gives you a color change from purple to blue as the reaction turns positive, whereas she used phenol red, which goes from pink to yellow. One thing I want to point out is that LAMP is quite inhibitor tolerant, so this is especially suitable for something for the quick lysis method that we're using. So LAMP is quite accepted in the diagnostic world, and this is one example of LAMP being applied to the forensic field, where the authors here you targeted the X and Y-linked amylogenin region and to do gender typing on dental samples. And they were using a turbidimeter to look for a positive change of 5 nanograms of DNA in about 30 minutes. So what I've done is, bless you, taken their primers and sort of reposition the primers to fit another loop primer, which is shown to improve reaction speed, but still targeting the Y-linked amylogenin region. Using the dye I just described and control male DNA, I compared the 2 primer sets. So on the left here, you have the no-gummy primers, and on the right, the redesigned primers. And what you're seeing down here is the range of DNA concentration used and the color change I noted along the timeline on the X here. So what you're seeing is that within 30 minutes, the redesigned primers at these concentrations changed color and completed the reaction, whereas the no-gummy primer still had the intermediate color change, so it's not quite there yet. And with the lower quantities, the redesigned primers were able to pick up 50 picograms of DNA in about 45 minutes. So now I have a lysis method. I wanted to make sure it's compatible with LAMP, so I took the lysate from the earlier slides here from these samples, quanted them, and they ran LAMP with the HMB indicator. So the lowest cell concentration here gave me 33 picograms of DNA, and that allowed amplification within 40 minutes. And with these higher concentrations, with these higher concentrations, the amplification completed in 30 minutes. So now I know that they are compatible with each other. And furthermore, I tested the specificity of my primer set. So I tested them against human female DNA and a range of DNA from other animal species. So what you're seeing here is that only the positive sample that contained male DNA turned blue, turned positive, while the remaining panel were negative. And that is confirmed by electrophoresis from the bioanalyzer, and that only the positive sample showed ladder-like structure, ladder-like band patterning. And like Tiffany has done, I can use hue analysis to look at the color change here to remove subjectivity. So purple gives me a hue value of about 200, and blue is anywhere from 150 and below. So you can see here clearly there's a distinction between the color change, and that I can quantitatively graph them. Again, very briefly on the image analysis process, we take a photo in a controlled lighting environment with a cell phone, and from there we can draw a line across the samples to compare the hue value against the background. And you can clearly see the distinction again from the positive to the negative samples. Or we can crop an area within that circle and get an average of the hue values, again, the distinction from the negative to the positive. And with that, I can do this at different time points and start to plot that along a time axis. So time's on the x here, hue value on the y-axis, and it's starting to look like a semi-real-time image here. So the blue traces here are male samples, and at different time points, and the peach-colored traces are female samples, and they remain negative as the negative samples did. And that allows me to start getting a time to positive value, or a TP value, which is similar to that of the CT value in qPCR. So to round this up, I did a little validation study which kind of mimics the Promega Casework Direct kit. So what they did was they took swab cuttings that had semen dilution down to 1 in 1,000, did the quick lysis, and they show compatibility with their quant kit as well as their STR kit. So what they showed was that you can see the STR and YSTR kit start to get allele dropout as the semen dilution gets to about 1 in 1,000. So very similarly, I did semen dilution, and I also included a female buccal swab to compare, deposited that on a swab, and did the Y screen process like I just described. So first of all, the quant showed that at the female sample is only female. There's no Y quant here. And then at the lower dilutions here, or the higher dilutions here, the quant is getting as low as 75 picogram per microliter. And this also showed that with the Plexer kit, it had no inhibition slide. That means the lysate is compatible with the Plexer kit. Next, moving on to the Y-screen lab results. What you're seeing here is the control samples, where the black trace here is the positive samples, and we have triplicated female swab cuttings up here in the pink, and that stayed negative throughout the 70 minutes with the negative control. And over here we have all the male swab cuttings, and they all turn positive within the 30 to 40 minute window. Again, look at the hue comparison with the negative here. And down here we have the broken down by dilution. So you can see here that unfortunately with the colorimetric flip method, I cannot distinguish the concentration of the semen dilutions. They all amplified within the 30 to 40 minute window. So it's, for this, in this case, it's more a qualitative answer. And so on the next slide, again, it just shows the lysate is compatible with the Fusion Astera kit, and that going down to 1 in 1,000 dilution, I was able to get a full profile from these swab cuttings. Lastly, this is just a summarized graph showing that in the gray traces here is the percent success for LAMP assay to identify these samples that contain male DNA, and that they correspond nicely with how well you can, how much, how it correlates with the Fusion Astera kit. So whenever LAMP picks it up, you can also identify it with a full profile in Fusion kits. So I have showed you I have an efficient lysis method, and that is compatible with our LAMP assay. And this assay is specific to human male DNA. And we have a we can quantitatively look at color to remove subjectivity, and the total process takes about an hour. And then the analyst from here can go on and decide if they want to do a DE or not. So in the future, we are going to looking at expanding this to co-extracting DNA and RNA. And like Tiffany said, we're moving on to doing more real-time studies and perhaps putting all this on a chip to automate everything. So I want to thank our sponsors, NIJ, and our collaborators at TGREX and NEB. And thank you for letting me speak at this platform. Again, I want to shout out, if you would like to test this out and have feedback for us, they are always welcome. So thank you. Thank you, Josh.
All right, thank you so much, Angie. We are going to move into our next presentation of the day. Our next presenter will be Miss Carrie Mays, and she'll be presenting on initial evaluation of the stability and persistence of mRNA and miRNA for body fluid identification in forensic samples. Carrie Mays is in the 5th year of her PhD in forensic science at Sam Houston State University. She received a National Institute of Justice Graduate Research Fellowship for her project investigating messenger RNA and microRNA in body fluid identification. She has also assisted in many other research projects, including human and cannabis identification using massively parallel sequencing and powder-free bone extraction methods. I'll switch everyone's layout now to Miss Carrie Mays' presentation. Carrie, if you'd like to go ahead and take it away.
All right, thank you, Josh. And I would also like to thank FDCOE for allowing me to present this. And I hope everyone on the East Coast is staying safe. This study was partially funded by a graduate research fellowship from NIJ, and the opinions, findings, and conclusions in this presentation are those of the authors and do not necessarily reflect those of NIJ. Getting right into it, body fluid identification isn't going to be the most important part of every case, but it can aid in the reconstruction of events, and the results of these tests can alter what charges the DA will pursue in a case. But these tests have issues. Many consume or dilute the sample, and not all body fluids are being tested for in all labs. The tests are also presumptive in nature and have varying levels of specificity as well as other limitations. In the cases of immunochromatographic cards, it can turn your lab into a bit of an impromptu game show where you have to turn to your neighbor and ask, do you see a line? Which of course can introduce bias. But as you heard from Tiffany and Anchi before, there's a lot of amazing research going into body fluid identification, including messenger RNA, microRNA, methylation patterns, and proteomics. But when I talk to analysts from labs, everyone is still a little bit hesitant, and I think this is for a few reasons. First of which being, do I have to? You guys have a lot going on. You just had to implement the expanded CODIS loci kits. You're either looking into or have implemented probabilistic genotyping, and you may have had to change around some things in your lab for legislation for sexual assault kit backlogs. So there's a lot going on. Next up, is the kit required? Is there something commercial or something in-house still going to be okay? Are you waiting for a shift to NPS? We all know that's a really powerful tool, and it'd be natural to wait for that paradigm shift. Last, are you sure it works? This is a big one, especially for RNA, which kind of has a reputation for degrading very quickly. But overall, more research is needed in order to get something concrete to the lab. And again, I would love to hear any of your feedback on this. But that's what we're here for, is to look into this research. In our lab, we decided to look at messenger RNA and microRNA. For messenger RNA, it can be co-extracted with DNA. It's going to be using instrumentation and techniques very similar to what's already in place in labs. There are 2 capillary electrophoresis-based assays already implemented in the Netherlands and at ESR. There have been numerous persistent studies looking at messenger RNA. And something I'm really looking forward to is SNP analysis using MPS, in which case we're actually able to look at SNPs in the transcript itself and do a level of fluid to source attribution. For microRNAs, they can also be co-extracted with DNA, and they also have been seen in DNA extracts as well, so it's something we would be able to go back to. Instrumentation and techniques are very similar, but it's still a little bit of the Wild West as far as consensus on body fluid specific markers, and several labs are still doing screenings on this. And there have been limited persistent studies looking at microRNAs. And while I lovingly think of them as the tardigrades of nucleic acids, their stability is mostly theoretical as far as environmentally challenged and forensic type samples. MicroRNAs are about 20 to 25 nucleotides long, which makes them a little tricky to work with. They're about the size of a traditional primer. Typically, you would use RT-qPCR using stem loop primers or polyadenylation. There is also a capillary electrophoresis-based assay, but it's still in its infancy. In the body, microRNAs are transcripted along with messenger RNA, and the expression profiles of these transcripts differ in terminally differentiated cells, so we can tell one body fluid from another. But while messenger RNA gets translated into protein, microRNA gets incorporated into a protein complex, which then can signal repression or degradation of messenger RNA. And we believe that it's this protein complex that's going to help stabilize microRNAs in the long term. But again, this hasn't really been tested, so it's something that we looked into. When we believe it's going to protect it like a hard candy shell. So the first thing we looked at was degradation over time. So we took blood and semen-stained cotton swatches, and we put one set in a lab drawer, so it's temperature, humidity controlled in the dark. And for the other set, we put them in the area on your left, where it has variable temperature and humidity, and it's exposed to natural light. And we took swatches on several different days. We extracted with the microRNA kit. We performed reverse transcription using the MyScript2 and the Quantitech kit. We performed PCR in duplicate with the MyScript Cyber Green PCR kit on a 7500, and we took 2 markers for each fluid. For blood, we took what was in red, and for semen, what was in green. We tried to choose an endogenous reference gene as well as a body fluid specific marker. Before we got started, we did perform some primer validations. We wanted to make sure that the data we got was comparable between messenger and microRNA. So we made sure that the same volume of extract and cDNA were going to each of the assays. We also looked at primer efficiency. Because there isn't much room for adjustment with microRNAs, we wanted to be sure that everything was working as it should. We also looked at melt curves to ensure that we are only amplifying one target. For data analysis, in most gene expression assays, you're going to be looking at the delta-delta CT values or the variation thereof. But in capillary electrophoresis assays, you're not necessarily looking at that strict relationship. And we also know that body fluids aren't homogeneous and some transcripts may be more protected in certain cell types. So overall, we were looking at a drop-in cycle number over time. We also monitored the temperature over time. As you can see, we had a bit of a roller coaster here in Texas. For our blood and our controlled conditions, so again, this is going to be humidity, temperature controlled in the dark. We saw a drop of about 7.5 cycles, or about 150 folds for messenger RNA. For microRNA, we saw a drop of 3 cycles, or about 8 folds. For messenger RNA, between day 4 and 60, we saw complete dropout, whereas for microRNA, we saw it was about 6 cycle drop. And again, you can see the overlay, just seeing the decrease between the two. And we know that humidity and temperature really affect messenger RNA. For semen in our controlled samples, we see a very similar pattern where messenger RNA is having a little bit of dropout, nothing substantial, and microRNA even less. We saw something a little more interesting in our experimental samples. So we can see, we can see that for our endogenous reference genes, which is going to be right here and right here, we're seeing maybe dropping off a little more quickly than our body fluid specific or a semen specific marker. And we think that may be due to the expression levels in the spermatozoa themselves. The endogenous reference gene aren't expressed as highly, and so we believe the semen-specific markers are going to be protected by those spermatozoa cells. And here you can see the overlay. And just looking at the fold change, we are seeing a very large difference between the messenger and the microRNAs. And again, for the semen, we're seeing something very interesting between the endogenous reference gene and the semen-specific gene. So overall, we're really looking forward to our later time points. It's suggesting that microRNAs may be stable, but even in these kind of insane conditions that got pretty hot in there, messenger RNA did hang around for a long time. Most of our degradation took place in the first 4 days. For our blood samples, we saw a very similar time frame as seen by a group in Florida. And again, we talked about those semen-specific transcripts being protected in the spermatozoa cells. The next thing we wanted to look at is laundry. Laundry is very tricky to talk about because there are so many variables, including laundry detergents, programmed cycles in your washing machine. There's lots of different variables, but it's something that we thought was very interesting. We do know that spermatozoa have been identified post-laundry and that some conventional body fluid ID techniques work. I will say that Dr. Kuhlstein had a very, very comprehensive look into this if you are interested. And she also looked at STR typing. And we also have seen some transfer of DNA being observed in some laundered items. So it was something that I would like to look into. So what we did was we took 3 different sources of blood and semen as well as an unstained cutting to look for potential transfer. We washed those in various settings. We had some with no other clothes, a cold setting, washed twice in a row in a cold setting and in a hot setting. And we also looked at these both air-dried and machine-dried. And I did use the infamous Tide Pod, which does contain enzymes. So even though I knew what I might be seeing, it does have me rethinking my detergent because I was able to visually see some of these stains right out of the washer. We did look at the semen stains as well under the crime light and orange goggles. However, we did not see any fluorescence. For our laundered samples, I know I said I wasn't looking at delta-delta CT values, but I couldn't help but wonder what it would, if it kept or not. So for the green, we did see the correct delta-delta CT values. For our yellow, we saw both markers, but they weren't necessarily in the correct ratios. In all instances where only one marker dropped out, it was the endogenous reference gene. So our orange is where we saw only our fluid-specific marker. And for red are instances where we saw a complete dropout, so both markers dropped out. For our blood samples, for microRNAs, both markers were there for every sample. And for our messenger RNA, we did observe some dropout, but we did at least see the fluid-specific marker in every instance. For our semen, again, microRNAs, we saw both markers in all instances. And for messenger RNA, we did see at least the fluid-specific marker in all but one sample. For our transfer, we were mostly looking just for our fluid-specific markers. We weren't as interested in the endogenous reference gene. So we did see lots of instances of transfer for our microRNAs, but not as much for our messenger RNA. So overall, just looking at our laundered items, we saw persistence of both messenger RNA and microRNAs through the laundry with and without other additional clothes, either air-dried or machine-dried, and most of what we saw is the body fluid-specific marker and the endogenous reference gene dropping out. With spermatozoa, they are very small, so they may be staying in that fabric. And we also saw transfer of both messenger and microRNAs to unstained areas, but was more common with microRNA. So in the future, as far as implementation, again, taking caution, but again, these are highly variable depending on many different variables, something like the length of your washing. So in the future, we are looking at massively parallel sequencing in challenging samples, so keep an eye for that. Again, I would like to thank FTCOE for allowing me to present this, and Houston State University, NIJ for funding my research, my lovely advisor, Dr. Sheree Houston, Dr. Rachel Houston, and Team DNA. And if you have any questions, I would love to hear from you. Please put them in the chat window. And thank you very much.
All right, thank you, Carrie. We're going to move into our next presenter of the day, Ms. Sarah Lum. We'll let her slides populate here. Ms. Sarah Lum will be presenting today on battling the backlog, a novel technology for rape kit analysis. Ms. Lum is a bioanalytical chemistry PhD candidate at the University of Notre Dame. After learning about the enormous backlog in rape kit processing, Sarah became passionate about utilizing her analytical skill set to design a patented technology to address this injustice. Sarah has been recognized with a 2017 Young Scientist Award, a U.S. National Institute of Justice Graduate Research Fellowship, and her work has been featured in the Analytical Scientist, Biola Magazine, and Notre Dame Magazine. She has traveled to crime labs across the U.S. to share her research and engage in conversation with law enforcement officers, crime lab analysts, and government officials to work together in battling the rape kit backlog. In addition to her research, Sarah is passionate about leadership, mentorship, and outreach. She served 2 terms as the president of the Association for Women in Science at Notre Dame, where she developed a STEM mentorship program for undergraduate women in STEM and chaired the 2016 inaugural Women in Science Regional Conference. And we'll switch everyone's layout now, and I would like to present to the microphone Miss Sarah Lum.
Thank you so much, Josh, for the introduction, and thank you to RTI and FTCOE for the opportunity to share some of this work. I am, as you said, from the University of Notre Dame. We are located northwest of Indiana, only a couple hours outside of Chicago, and I'm excited to share about the technology that we're developing for rape kit analysis. Now, before we start our engines this morning, I wanted to give you a brief roadmap of where I'd like to take us in the next 10 to 15 minutes. I'll begin with some background information, discuss my motivation behind this work, as well as the current methods of rape kit analysis in crime labs across the U.S. We'll then discuss the sample collection and preparation procedures and our separation using a technique called capillary zone electrophoresis. This would be separation of the male from female DNA from rape kits. We'll then collect the purified aliquots of male and female DNA with an automated fraction collector and move on to real-time or quantitative PCR amplification with further downstream analysis. So the motivation behind this work was when I learned about the tens of thousands to 500,000 untested sexual assault kits in the U.S. As Renchi said, the current estimate is about 200,000 or more. And a sexual assault kit is a collection of swabs or smears or hairs, anything from the body of the female victim that could potentially contain DNA from the male perpetrator. Now, since the vast majority of sexual assault is a male perpetrator on a female victim, that's what I'll be referring to for the remainder of this talk. Currently, there's a 100-day turnaround time from the collection of one of these kits to it being sent to a crime lab for analysis. And since estimates of 50% or greater of rapists are repeat offenders, that's 3 months of time where they are able to walk free, potentially committing the same crimes to countless other victims. This is a huge problem in the U.S. and abroad, and it's something that needs to be addressed with policy and communication, but also with better technology and better methods of processing. So the current method is called differential extraction, and again, that's the separation of just cells based off the differential stability for their membranes. We're looking to separate out our male sperm cells from our female epithelial cells. And we're looking specifically at the male sperm cells because if we can get DNA from that, we can prove that there's a sexual nature to the crime. So the first thing you'll notice is your male sperm cells are very small, about 5 microns compared to the 40 to 60 micron epithelial cells. The second is the sperm have a much stronger, more robust membrane. It's a lot harder to break open, whereas the female epithelial cells are very fragile. And if you take a swab from your cheek, most of those will be lifed open. The other thing is that the sperm is a very small portion of the sample. Since these swabs are collected from the body of the female victim, most of the DNA we're going to get is from the female. And there's estimates of 1 to 10, 1 to 50, 1 to 100 or greater the sperm to epithelial cells on that swab. So if we were to take that swab and analyze it directly, you're going to get an overwhelmingly female profile. We already know who the victim is. We're trying to find out who the male perpetrator is, so we need to separate these out in order to get a male profile. So differential extraction, you begin with your sample collection. You're going to do a mild buffer extraction first, and then you're going to go ahead and centrifuge that sample, and that will give you your light cells or smaller particles on top of your tube, and your whole cells or larger cellular debris at the bottom, and that gives you your epithelial cell DNA. You can repeat that process with a centrifuge, pellet, and wash with a harsher buffer extraction to target your sperm cell DNA. And in different crime labs, you'll repeat this process multiple times. You'll incubate for, again, multiple hours, different temperatures, different dispersions, different pH. All you're looking for is getting cells as much separated as possible before you do your PCR amplification and your SPR analysis. So differential extraction is good in that it does selectively isolate the male DNA prior to profiling. And again, that's essential to get any male profile. It's a fairly simple procedure. You can teach a lab tech to do that fairly easily. And it's reasonably effective in at least producing a primarily male fraction. So you will never be able to get 100% separation using this method, but it could get you good enough to get a male profile. The problems and the challenges with it is the extraction could take 3 hours or more. Different crime runs incubated overnight, and they work on that same kit for a week or a couple weeks. It's very labor intensive. So all of those centrifuge, pellet, and wash steps is an analyst actually has to be pipetting those samples from place to place, and that also introduces the risk for sample contamination or sample loss. These are forensic samples, so you have a limited amount of available evidence. You can't allow that risk for contamination. So what I'm proposing is a novel method that uses CE for the separation. We'll do the same sample collection, but I'm going to do just one buffer extraction. And I'm going to use a buffer that doesn't harm the cells so that I can rely on the capillary zone electrophoresis for the separation of your male and female cells. And I'll actually be separating intact cells and collecting them on the other end with an automated fraction collector that was also developed in-house. So CE uses a small sample size. It's a non-destructive technique, so everything can be collected at the end. And it has very limited analyst interaction with the sample. So we don't need to worry about that sample contamination or sample loss with sample transfer steps. You'll inject it into the CE, it'll separate it, it'll put it right onto your 96-well plate, and that is standard in the vast majority of crime labs that I've been in communication with, so it fits well within their workflow. And then we'll do our PCR amplification and STR analysis. So that leads us to our sample collection and preparation. For the sexual assault kits, I have two types of samples that I'll need. The first is the epithelial cell, so the same cell that lines the esophagus, mouth, and vagina are these guys here. They are about 40 to 60 micrometers in size, and then negatively charged at physiological pH, but that exact charge is going to vary very greatly depending on the person, depending on the diet, depending on the time of day. What I can do is get some volunteers from Notre Dame, wonderful graduate students or people who let me speak in their group meetings, and I'll take a cheek swab, and I'll just get 5 to 10 seconds on either side of your cheek. I'll put that back in the tube and air dry it overnight in the hood, and that'll give me my epithelial cell DNA. The spermatozoa are these guys here, and what I'm interested in is, again, the DNA in the nucleus. It is protected by the acrosome, which makes up the head of the sperm cell, and you've got the mitochondria, which is the powerhouse for its little gurney, and the tail, which actually falls off fairly easily even with mild agitation. So I'm mostly interested in the head piece, and it's covered by a 20 to 60 nanometer glycoprolate layer, which gives it a negative 16, negative 20 millivolt charge. These guys were a little bit harder to collect, but I have a wonderful collaborator from UIUC who gives me the semen samples for the sperm cell. So we've got our samples ready to hit the road with our separation using capillary zone electrophoresis. So CZE is a technique that uses a long, thin, hollow tube, very similar to the size of a strand of hair. And it's made of silica, so if I were to flush a base like NaOH through it, I can get a negative stationary layer, flush a buffer through it, and apply a voltage to either end. You get a positive mobile phase that creates your electroosmotic flow, and that is the force that will actually drive the separation. So here we are separating out samples by the unique charge and size ratio, with your smallest, most positively charged particles migrating out first, followed by neutral, followed by your negatively charged particles. What we'll expect for this separation is our sperm cells to migrate out first, followed by any lysed epithelial cells. Again, those are very fragile, so we do expect a good amount of lysis, followed by any whole epithelial cells if there are any left in the solution. So this is the instrument that we designed, and you'll see here your sample starts in the injection block. You can do either an electrokinetic or a pressure injection. And the samples separate across this capillary here by size and charge. We'll be able to detect it with light scatters using a 532 nanometer laser. So anything that passes through that beam I can detect. The sample is amplified by what's called an avalanche photodiode and sent to the computer for further analysis. And this here is the actual interface. So you'll see the laser is actually coming from here and going across. Your capillary is going to the top of its teeth. And the APD, which sends your signal to the back. On this side is a diagram demonstrating the sheath flow cuvette, and that is what focuses the sample directly on plate scheme. If you look through this objective lens, you can actually see the capillary inside the cuvette, the laser passing through, and you'll see bright flashes of light as the sperm or epithelial cells or another debris passes through the capillary. So now we're ready for collection. So we've determined the migration time of our sperm and our epithelial cells using the light scatter. And so we've developed a second instrument that's very similar. So you'll see the injection block here. And again, a pressure electrokinetic injection separation through the capillary. The only difference is this right here. Instead of the laser detection, we are collecting it into a standard 96 well plate. And this is a translational stage that actually moves so that your sample can be deposited into the individual well. And CE uses a very small sample flow, so the sheath reservoir will go ahead and deposit that sample. So this is how our plate will work. We'll go ahead and deposit a sample into one well. The translational stage will move the plate in a serpentine-like fashion, depositing samples in each of those wells. So we can separate the samples from CE and then collect those purified fractions at our set intervals. And now we've got a microtype plate that's ready for analysis. So what you'll see here is a plate diagram, and we can put our controls in that top well, positive and negative controls, and then we can inject the semen sample, we could inject the epithelial sample, allow that to separate out through, and we'll expect 1 or 2 of those wells to contain our sample. Now we're ready for real-time PCR amplification. And the way that works, bear with me if you are very familiar with the process, but I wasn't sure. It sounds like we've got a broad audience. So we're looking at an amplification of DNA. It's a 3-step procedure. You want to denature your DNA template. Go ahead and anneal your primers. And I'm looking at a Y chromosome specific primer for now, just so I can tell where male reading. We'll anneal those, and then we'll extend the DNA, and that'll give us an exact copy of that DNA. So amplification of a specific sequence with extraordinary high fidelity. So forensically speaking, what we're looking for are these specific sequences called 20-core STR loci, short tandem repeats. Those are repeats of 2 to 13 nucleotides repeated hundreds of times in a row on a strand of DNA. And so the chances of any 2 people in the world having the exact number of repeats in each of these 20 regions is significantly less than the number of people who have ever lived on the planet. So we can be 99.9% certain that if someone matches the crime scene DNA, that is our, or that suspect matches the crime scene DNA, it's the same person. And so what we're doing is real-time PCR, so there's just a fluorescent tag at the end of each of those copies. We can monitor it in real time. What we'll be looking at next are these plots where you've got your cycle number, which is your time on your x-axis, and your y-axis is your relative fluorescent units, which is signal. No template should not give us a signal. We're looking for when the sample crosses this threshold value. We'll call that the concentration, the T value. So if we throw in DNA-free water, that's a blank control in our top row, we shouldn't see any signal. Then with our female epithelial control, since we're looking at a Y chromosome primer, we expect the male epithelial cells to give a signal, and same with our semen control. The 10x, 100x, and 1000x dilutions is equivalent to about 600, 60, and 6 cells. When we go ahead and toss the semen on the E instrument and fraction collect, we'll see sperm cells. We've verified it by hemocytometer, intact sperm cells coming out in about 10, and that gives us about 6.5 minutes to 7 minutes of migration time. And then we also see some epithelial cells in the semen, since the urethra is lined with epithelium. That'll come out in C11. We verified that is the male epithelial cells by injecting just the male epithelial, and since those can lyse and give us different sizes and shapes, we do see a little bit wider spread in that. But we're getting a separation of the sperm cells and the epithelial cells in these different solutions, which is exactly what we're looking for. So in conclusion, we've created a simple and effective sample buffer preparation, which does not lyse our sample, performed a 10-minute separation of the sperm from epithelial cells using CE, collected those guys in a 96-well plate, which fits into the workflow of a crime lab. And all this demonstrates that this is a potential instrument that could be used, promising technology for rape kit analysis. With that, briefly, I'll just thank my lab group and Dr. Ashley Hall, University of Illinois at Chicago, for her samples, as well as the NIJ for their support, and a bunch of other crime labs and other universities that we've spoken with to give us the resources and advice to do this work. I will be happy to take any questions at the designated time. Thank you so much for your time.
All right, thank you, Sarah. We are going to switch to our next presenter. Our 5th presenter of today is Mr. Fabio Oldani. We're going to let his slides populate here. Again, this is our first day of our National Forensic Science Week. Dr. Fabio Oldani will be presenting on analysis of microhaplotype markers on massively parallel sequencing platform. Dr. Oldani holds a Bachelor's of Science in Biology and a Master's of Science in Microbiology and Genetics from the University of Pavia, Italy, and also a Master's in Forensic Science from King's College of London. He received his PhD in Forensic Genetics from the University of Lausanne in Switzerland in 2017, where he worked on both the first development application of DIP STI markers for mixture deconvolution and touch DNA. Since October 2017, he has worked on He has been working on postdoctoral research on massively parallel sequencing of microhaplotypes at the Forensic Molecular Biology Laboratory, Department of Forensic Sciences at the George Washington University in the United States. We'll go ahead and switch everyone's layout now. And Fabio, if you'd like to take over the microphone.
Thank you, Josh, for the kind introduction, and hello, everyone. First of all, I'd like to really thank the Forensic Technology Center of Excellence and the RTI for hosting this National Forensic Science Week webinar and for the opportunity to present the latest results on some MPS projects that were running at the George Washington University. So in this webinar, we talk about the analysis of microhaplotypes on massively parallel sequencing platform. Microhaplotypes were recently proposed as a novel type of genetic marker to complement SDR analysis. We know that the concept of microhaps for forensic purposes was first introduced by Professor Kincaid. What is a microhaplotype? Well, it can be defined as a locus of less than 300 nucleotides in size, which consists of 2 or more closely linked SNPs that can be associated in 3 or more allelic combinations. We know that Sanger sequencing is the gold standard sequencing method, but the main drawback is that it doesn't allow determining the cis-trans relationship among individual SNPs. On the contrary, the emerging massively parallel sequencing technology has the ability to distinguish parental haplotypes with one specific locus by clonal sequencing of each DNA strand, and thus allowing to determine unambiguously the phase of all SNP loci. Here is an extensive list of papers which describe the exploratory and discovery phases of microhaps. Initial studies conducted by the KIT lab were focused on the search of markers across the whole human genome. And since microhaps are multi-SNP loci, and we know that SNPs are effective markers for ancestral prediction, there is no denying that a lot of attention was also drawn to the potential using these markers for ancestry inference. And the plot over here shows you an example of a microhaplotype locus that displays different alleles with variable frequency among a global set of populations, thus making this locus promising for biogeographic ancestry inference. Now, since MPS technology is available for forensic analysis, other research groups have started developing new sets of markers on different MPS platforms, and here is the list of papers that were recently published. So, what are the advantages and disadvantages of microhaps compared to SDRs? Well, one of the main advantages is that there is no preferential amplification for short alleles. There is also no started peak, which is a huge issue for the analysis of SDRs, especially when the alleles or the minor contributor fall in started range position of the main alleles. Then the mutation rate of SNPs is much lower than SDRs, and ancestral information can also be provided. On the other hand, microhaps have also some disadvantages. They have a reduced number of alleles compared to SDRs, which are more polymorphic loci, and require using a large panel of markers to obtain a probability value of DNA evidence comparable to SDRs. Right, on the right-hand side, you can see an overview of the current and potential applications of microhaps. These markers can be used for ancestry prediction, mixture deconvolution. They can also be implemented on probabilistic genotyping software for the analysis of complex DNA mixtures. And by taking advantage of the fact that SNPs have low mutation rate, microhaps can also be potentially used in relationship testing, in human and missing person identification, clinical applications, and non-human DNA analysis. However, in this webinar, I will focus your attention only on mixture detection and deconvolution, implementation on probabilistic genotyping software, and ancestry prediction. Right, to date, the KIT lab has characterized approximately 200 microhaps, and a few more were recently developed by other research groups. The paper cited over here represents the latest paper which evaluated a large number of markers across a global set of populations. So basically, from this article, we selected 74 loci, which showed high effective number of alleles, which represents an important statistics for mixture deconvolution, high level of informativeness across different populations, which represents a key factor for ancestral inheritance, and different chromosome position. And then in collaboration with Thermo Fisher, we built up a multiplex assay of 74 loci that we implemented on the latest Ion Torrent platform. So for those who are not familiar with the MPS technology, here is a slide which covers different steps of the full MPS workflow. So we start with a standard PCR reaction, and then we proceed with a manual or automated library preparation, which includes ligation, barcoding, purification, quantification, and normalization of libraries, and preparation of the pooled library. Then the templating step is performed by the Ion Chef system, which uses a specific emotion PCR process to load the pooled libraries onto the Ion chip, while the actual sequencing occurs into the S5 instrument, and raw data is analyzed through an MPS pipeline. So how the MPS data look like? Well, alleles are represented as histograms. The sequence is shown on the x-axis and the coverage on the y-axis. So basically the MPS output is pretty much similar to the output from the conventional CE platform. Now, the first question that we wanted to address was to determine the detection limit of the assay. Right, we performed a sensitivity study, and then we compared the results to CE STR typing. We selected 3 DNA samples and prepared series dilutions from 2 nanograms to 25 picograms of input DNA. Each DNA sample was amplified in triplicate using the global file STR kit containing 24 STR markers that were run on the 3130C system, and the same samples were then amplified using a 74-plex Microhaps assay that was run on the S5 instrument. Right, for STRs, full profile was obtained up to 100 picograms of input DNA, while allele locus dropouts were observed at lower DNA input. For Microhaps, our analysis indicated that we could recover a full profile at 50 picograms of input DNA, while some alleles drop out at 25 picograms. Now, the first application I'm going to present to you is a mixture deconvolution. So how to test the efficiency of the 74-plex assay? To address such a question, we prepared a first set of mixtures and then tested the performance of the assay to detect one or more minor contributors. We simulated 2% DNA mixtures at different ratios using both 1 nanogram and 10 nanograms of DNA, and also 3, 4, and 5% DNA mixtures using only 1 nanogram of DNA. And overall, for 2% mixtures, we could recover a full microhaplotype profile 20:1 ratio using 10 nanograms of DNA, whereas a few alleles dropout were observed at 1 nanogram at the same ratio, and more loss dropout more at higher mixture ratios. For 3, 4, and 5% mixtures, we basically obtained a full profile at the mixture ratios that we tested. So now let's have a closer look at the performance of Microhaps in mixture analysis. So to make things more understandable, I'm going to show you the performance of only 2 out of the 74 loci, where the minor DNA was heterozygous at both markers. So here we have a 2% mixture at 40:1 ratio. And as you can see, we obtain a full and partial genotype for these 2 markers using 10 nanograms of input of DNA, which mimics to some extent the amount of DNA recovered from a typical sexual assault sample. And overall, a total 36 alleles were reported for the minor DNA. Below, you see the same 2% mixture amplified using 1 nanogram of input of DNA. And we can observe that 1 locus dropout and 1 allele at the other locus dropout as well. So even if we observe an uneven performance across the 74 loci, a total 26 alleles were reported for the minor DNA. Here is an example of a 3% mixture, a 10:1:1 ratio amplified using 1 nanogram input of DNA. And for this mixed sample, we could report both alleles at the 2 loci for the minor DNA 1 and 2, although minor DNA 2 shared 1 allele with a major donor. And it's also worth pointing out that for Microhaps, the signal from the minor donor was found higher than the signal for SDR markers, for which we couldn't deconvolute the profile. However, the interpretation of these kind of mixtures can be significantly improved and simplified using probabilistic genotyping software. So in collaboration with Dr. Benschop from the Netherlands Forensic Institute, we developed a new version of LMX Studio to analyze Microhap data. So LMX Studio is a semi-continuous software which requires the user to input the genotype or mixture and the reference samples, set the appropriate propositions for the prosecution and defense, and then the software provides the user with a likelihood ratio value. In this particular case example, the LR value is calculated for the two minor DNA contributors, with both in the order of approximately 1 billion. Right, now that we've seen the performance of Microhaps in mixture analysis, the last application I'd like to talk to you about is ancestry prediction. To assess the potential of the MPS panel 74 losses to infer the ancestry of individuals, we selected and genotyped individuals from different American population groups: African Americans, European Americans, East Asian Americans, and Southwest Hispanics. We collected and generated allele frequencies for the 74 markers of each population group. Now, how to express the ancestry inference? Well, it can be expressed in terms of random match probability, or RMP, and can be calculated using the allele frequencies generated for each population group. And to evaluate the efficiency of the assay, we selected a set of 9 unknown testing samples representative of each population group, and then we calculated the RMP for the 4 corresponding populations. And interestingly, the RMP was found significantly higher in all populations where individuals self-identified as such. And overall, the RMP values were found in the order of 10 to the power minus 60. In addition, we used the phased inferred allele frequency for the 74 loci uploaded on Alfred, which stands for the allele frequency database, to infer the biogeographic ancestry of some unknown testing samples. In particular, we used allele frequencies reported across 26 populations, representative of the 4 major population clusters: Africa, Europe, Asia, and America. And we selected 20 unknown individuals who self-identified as African Americans, European Americans, East Asian Americans, and Southwest Hispanics. And then we tested the efficiency of the 74-plex panel to predict the origin of these samples. Right, for African, European, and East Asian Americans, the log 10 of the RMP was much higher in the relevant population tested, while for Hispanics, the log 10 of the RMP was equally high within European and American populations, thus confirming the mixed level of this population group. Finally, when testing one sample of Native American origin, particularly Mexican Piedmont sample, the log 10 of the RMP was found higher in the American populations, proving that our MPS assay can correctly predict the ancestry of unknown individuals. Right, to sum up, the assay that we developed is sensitive up to 50 picograms of input DNA. It's efficient in mixture detection and deconvolution, can be further implemented on probabilistic genotyping software for the analysis of complex DNA mixtures, and can also provide ancestry information. Our current work is focused on a more comprehensive study on DNA mixtures and comparison to MPS STR typing in addition to CE STR typing, but also performing a genotyping study of a large set of global population samples and structural analysis. We're also evaluating some continuous and semi-continuous probabilistic genotyping software for the analysis of complex mixtures. And finally, we're also evaluating the potential use of Microhaps for investigative purposes. Right, to conclude, I'd like to really thank again the Forensic Technology Center of Excellence and the RTI for the opportunity to present on this platform. Also the National Institute of Justice and the Swiss National Science Foundation for the financial support provided. HT Group from Thermo Fisher, Professor Ken Kidd, our key collaborator, as well as all GW master students involved in our project, and last but not least, Professor Bodini. Thanks very much for your attention.
All right, thank you so much, Fabio. We're going to move into our last presenter of the day. Thank you again for all of our presenters so far, and thank you for all the questions from the audience. We'll get to those shortly. Our last presenter of the day is Ms. Zinat Zalahuddin. Her presentation will be on predictive modeling of nose shape variations on the basis of known genetic associations. Ms. Salahuddin is a visiting researcher at Penn State University in Mark Shriver's laboratory. She is doing her PhD in forensic sciences at the Department of Forensic Science in the University of Health Services at Lahore, Pakistan, under the supervision of Dr. Ala Rakha. She followed her PhD coursework major in forensic biology, particularly about advances in forensic molecular biology and genetics, with her project being about predictive modeling of nose shape variation on basis of known genetic associations. She is currently working on genetic characterization of nose among Pakistani population and is working as a project coordinator in facial capture and collection database. We'll switch everyone's layout now to Ms. Zinat Salahuddin's presentation. Zinat, if you'd like to take it away from here.
Thank you, Josh, for introduction. I would like to thank Forensic Technology Center of Excellence and RTI for providing a platform to the young scientists. Questions are welcome. So today we will talk about predictive modeling of nose shape variations on the basis of known genetic association. As you know, phenotypic variation in human facial morphology is substantial and plays a very important role in identification, especially in faces where crimes are often committed and there are no surviving witnesses providing important information for facial characteristics, and it is also useful for missing person identification. So the advancement in science enable us to work on facial morphology and genetic methods to facilitate us to provide phenotyping. So before jumping into the topic, let's have an overview of a DNA phenotyping. What is DNA phenotyping? Basically, it is a process of predicting an observable traits using genotypic information collected from genotype or DNA sequencing. And this term is also known as molecular photo-fitting, which refers to the prediction of a person's physical appearance and biogeographical ancestry for forensic purposes. So there are basically two types of molecular photo-fitting. One is indirect molecular photo-fitting, that is phenotypic inferences drawn from the genetic ancestry estimates only. And second is direct molecular photo-fitting, which is a phenotypic inference drawn from genetic test or a functional loci. So the question is, why would molecular photo-fitting will be useful in forensics? When we have both types of a DNA evidence and references based on a DNA, we use different contemporary ID markers such as SDRs. When we have facial images as reference and evidence, we use biometric methods to compare video and still evidentiary photos to reference photos. But when we have DNA as evidence and reference as facial image, or vice versa, predictive modeling, predictive biometrics is useful on evidential DNA to generate new investigational leads. Indeed, forensics DNA phenotyping, which acts as a biological or a DNA witness, is yet another step in the evolution of investigative tools like fingerprinting, handwriting analysis, polygraph, DNA typing that enhance the efficiency with which crimes are solved. So in recent years, phenotypic science has grown exponentially. Current studies conducted to enhance the accuracy and reliability of our forensic casework have shown that phenotyping can predict externally visible characters with high level of accuracy. The predictive value of phenotyping is presented by the discovery rate like hair color, eye color, freckling, dimpling, cleft lip or palate, their chronological age, the identification of earlobe attachment genes, adult height, male pattern baldness, dominant headedness, and scalp hair, roll number, and direction. So recent GWAS studies open up so many avenues to broaden up research, but there are areas which still need to be explored. In 2012, San Liu first conducted a genome-wide association study for facial shape phenotypes in multiple discovery and replication cohorts on European descendants and identified few independent genetic loci associated with different facial phenotypes, suggesting the involvement of 5 major candidate genes that are PRDM16, PAX3, TP63, and C5 and COL17AI in the determination of a human face. Again, in 2012, Peter Noster aimed to identify genetic variations associated with normal facial variations in population based on 15-year-olds from Avalon longitudinal studies of parents and children, and identify PAX3 gene associated with nasal position. In 2016, Adhikari and co-workers reported a genome-wide association scan for facial features in Latin American. They evaluated 14 traits on an ordinal scale and found significant association in 4 genomic regions of columnar inclination, nose breadth, nose wing breadth, and also identify SNPs to chin prostitution. So in 2016, again, Cole and his colleagues tested for genetic association of facial shape and size of a phenotype derived from the 3D imaging and automated landmarking of standard facial morphometric points in African population. SNPs within the genes were associated with a measure of the facial size in both the GWAS and replication cohorts that passed very stringent genome-wide significance tests adjusted for multiple testing of 34 correlated traits. In 2017, Lee and his cohort workers performed GWAS on European ancestry and found associations with mid-face height and upper lip height. In 2018, another huge genome-wide study of Peter Klaas and his colleagues performed GWAS and found 15 loci including novel association which affect multiple facial traits. And in 2018, Cha and co-workers performed Korean GWAS for facial traits and found novel loci related to the facial frontal contour, eye shape, and nose shape. So the question is, what are the factors involved behind the genotype-phenotype analysis? Facial feature gene discovery depends on various things like craniofacial diseases, mapping, normal range mappings, and sex-dependent mapping on animal models, gene expression, and population and functional genomics, which in turn helps to develop facial models depending on the SNP genotyping and the self-reported factors. So, moving on to my current research project, I'm working on Pakistani population. Pakistan is a hub of migration and have various climates throughout the country. In the north, we have China here, and like in the east here we have India. In west we have Afghanistan and Iran, and in south we have Arabian Sea. So along with diverse lineages of Eurasian, South Asian, African, Austronesian, Mongolite population, few American populations. So my project has basically 2 basic aims. One is to characterize genetic basis of nasal shape, and other one is to develop a simple genotyping assay to classify nasal phenotype. So given great importance of finding these genes that have been discovered in European population, we recruited 300 individuals from Punjabi population, collecting their blood for DNA, their 2D and 3D photographs, self-reporting ancestry, along with their demographic and anthropological information. So as you see in the map, marks show the birthplace of the subjects. So for the perception study, we're going to divide nose shapes into 3 broader categories that are leptorine, mesorine, and platurine. So as for methodology is concerned, for analyzing sample, we use panel of 900,000 SNPs using candidate gene approach for 57 male samples, evaluating nose shape pieces for genetic pieces, their age, their BMI, place of birth, their caste. So for 3D image processing, we apply geometric morphometric approach. So for each face, we have original 3D photographs, which is trimmed and then reflected, putting anthropological marks, which has 7,150 indices, and then it is remapped and reflected to superimpose to summarize into an so we have projected phase. And from here, we're extracting nodes from the 3D phase, which has 2017 node indices. Here are a few examples of a remapped 3D faces from Pakistani population. You can prominently see the difference in facial phenotype and nasal variation. And here are a few genes and SNPs which are discovered, like PAX3, IFR6, RA1, and intergenic genes, which was found in Pakistani population, and they are consistent with other European studies. General trends in the results shows that age is a significant predictor of nose PCI, BMI is significant predictor of nose PC3, age and BMI is significant for nose PC2, and caste, ethnic groups is significant predictor of nose PC4, while genetic PC3 and PC4 and SNPs are significant for nose PC5. So the future directions include facial averages, face, nose segments, and consensus for male and female, which include testing the rest of the candidate gene splits in a larger sample, setting up a genotyping assay, and performing a Pakistani GWAS for nose and other facial features in even larger sample, linkage analysis in families, and testing genes uncovered and candidate genes for signatures of the selection. So in the end, I would like to thank my team from University of Health Sciences Lahore and Penn State University, especially Mark Shriver and Dr. Alaraka for this opinion. I would like to again extend my thanks and gratitude to Forensic Technology Center of Excellence for providing me such a wonderful opportunity. Thanks. I would love to have
Thank you so much,
from you.
Ms. Salahuddin. We are going to go ahead and move into our Q&A session for today. We did have quite a few questions come in, so we're just going to jump right into it. We'll pose our first question to Ms. Tiffany Lane. How much of a sample was needed for your analysis, and is this method destructive?
Yes, thank you for that question. For this assay, you only need to use 1 microliter of your extract from your sample. And unfortunately, yes, it is destructive. However, in the extraction method, you can not only extract out RNA, but there are the DNA and RNA kits you can extract out, so that way you still have your sample for downstream profiling.
Okay. Next question comes in for Anchi Sui, asking, have any inhibitors been identified for this test?
All right. So the samples we've tested have been buccal swabs and semen on a swab. So these samples have not been identified with any inhibitors, and the Plexer data that I showed earlier had no inhibition flag. So at least for buccal and semen, we know there are no inhibitors, and there definitely isn't any inhibitor from any of the reagents we used. I mentioned that LAMP is good for, and it's tolerant, it's because it's quite well published that it can tolerate things like calcium. So if you're working with bone, calcium is less inhibiting to LAMP compared to PCR. And there's also publications on LAMP being more tolerant to salts, to blood, to urine, and stuff like that. So I would say I haven't seen any, but as we start to test more realistic forensic samples, then we might come across it.
All right, thank you so much. Next question comes in for Ms. Mays. Rebecca's asking, how would this be applied to asporatic individuals?
Hi, Rebecca. Great question. So during the screening for both messenger and microRNA markers, labs were very, very careful in choosing to make sure that there are markers available for both for azoospermatic individuals. So I did not choose them in my study, but they are available, and in most assays, you're going to see that diversity where several markers are available for one fluid. such as KLK3 or TGM4 for messenger RNA, or MIR10 for microRNA. So there are markers that are specific for semen, but not spermatozoa themselves.
All right, thank you. Next question we'll pose to Ms. Sarah Lum. Mark Miller's asking, do you have clogging problems with the capillaries?
Sure, thank you for the question. No, I'm using capillaries of 100 micrometer inner diameter, and we have not seen any clogging issues with that.
All right, thank you, Sarah. Next question came in from Rebecca. She's asking Fabio, is the mixture deconvolution software available separate from the S5 software for use with other MPS instrumentation in regards to the LR mixture software?
Right, that's a good question, and the answer is very easy. Yes, the probabilistic genotyping software that we developed in collaboration with Dr. Benshop can take any type of data from any platform. It just requires the allele frequencies for the all loci that are included in the panel.
All right, thank you. The next question we'll throw out there to Ms. Zinat Salahuddin. Zinat, you had mentioned that your primary focus was on the Pakistani population due to the wide range of immigration and movement. Do you have any plans on doing the same type of study in the United States?
Yeah, sure, definitely. We will work on that population as well. But currently, I'm working on Pakistani population, and my future plan is to go back to Pakistan and collect more samples from other part of the country and come back here to work on that. And definitely, we will include samples from
All right, great.
United
Thank you so
States
much.
as
We got
well.
a couple questions left. I'll throw this one back to Tiffany Lane. You kind of addressed what the minimal RNA input requirement for accurate result was, but also did you test your method in mock casework samples?
Thank you for that question again. Yes, we have tested some sensitivity with this assay in the different fluids. We have gotten down to approximately 312 picograms per microliter that we can input into this assay and get accurate results from. And we are optimizing this further to see if we can get that even lower. For blood, we've gotten down to 156 picograms per microliter, but the same thing, we're still optimizing that to see how low we can go. And then the second question was, have we tested mock case work samples yet? We are in the midst of doing that. As we're finishing up the optimization for some of the other body fluids, that's our next step, is to test those and show what it looks like against some of those types of mock samples that crime scene investigators and forensic scientists would see.
All right, thank you. Next question we have is actually a question from me. I'm asking Angie and Tiffany, you can chime in as well. I'm noticing in your presentation you were using a smartphone. Do you have any kind of requirements for the smartphone, such as operating systems or specific apps or programs that might be needed?
Well, the phone that we're using has been optimized for the box, so the size of the phone where the camera locates is important. So we use only one phone to take a picture with our box. But we have looked into other phones. Initially we were using the iPhone, but eventually we decided on the specific phone because it works well with the size and the dimensions.
All right, thank you. Last question we have we'll pose to Ms. Mays. Joseph Warren is asking, I've been interested in looking at degradation of DNA and RNA for information on time-sense intercourse. What are your thoughts on that?
Hi, Joseph. That is a very interesting question. I think it's also, just like the laundry, it's going to be very tricky. I mean, from internal flora, as far as diets of both people can really change the environment of where, like in the vaginal cavity. So I am not, as far as DNA, I don't know. I haven't really looked at any type of postcoital samples at this time. I am not sure you would ever be able to get something reliably just because of the natural variation between people. But again, it's something I haven't looked into, but it is an interesting thought.
All right, thank you so much. We are going to go ahead and move into our closing. Obviously, the first thing we would like to do is to thank all of our presenters today. Thank you so much for all of your hard work and all of the research that you are conducting. We're very excited about all of your careers and you moving forward. So thank you for taking the time to present today and be the first part of our National Forensic Science Week. We would also obviously like to thank all of our attendees for joining us today. Thank you for the great questions. These webinars would simply not be possible without your attendance and support. Before you leave today's webinar, we always ask that you participate in our short survey. You'll find that there in the middle of your screen simply by clicking here. A new window will open up for you and you'll be able to take our survey. This feedback is valuable information that we use and that also all of our presenters will be able to take a look at as well in regards to your opinion on their research and their presentations. We are going to be putting there in the chat pod some information here shortly about upcoming events for this week. We do have 2 more webinars that are scheduled for this week. We have one that will be on tomorrow, the 19th, titled Novel Forensic Chemistry Research from Early Career Scientists. It will start at 10 a.m. Eastern Time again, so be sure to check us out for that. It'll be pretty much the same format as this. And then we have a fantastic presentation on Friday with 3 laboratory directors making a forensic scientist, a recipe for success, where they will go over what early career forensic scientists should see and expect when they are coming into the field. So be sure to take a look at our website. Julian's posting all of that information there in the chat pod now, and make sure to come and join us for the rest of our webinars this week. You will see there in the bottom left-hand corner of your screen, we do have a few presentations that we were able to disseminate out to you, our audience, today. So make sure you download those before you leave. And again, thank you to all of our presenters for your research and your presentations and your time today. And thank you to all of our participants for joining us. We hope to see you back for the rest of the week. And we hope everyone has a great rest of your afternoon.